Hey, did you ever wonder how to identify non-coding genetic variations that affect TF binding? Are you interested in detecting the regulatory impact of such SNPs? Then you're right here. In the following few minutes, I'm gonna explain our new statistical approach which can help you to answer exactly these questions. Genetic variations such as SNPs or short insertions or deletion occur more often in non-coding DNA regions than in coding regions. And in this pie chart, you can see that actually 88% of the genetic variations which are associated to diseases or traits are overlapped with the non-coding DNA region, and out of these, 31% overlap with the binding site, which is occupied by a transcription factor. And based on this observation, one can argue that these non-coding genetic variations can be functional. In this talk, I denote SNPs which affect the binding behavior of a TF as regulatory SNPs. And regulatory SNPs can lead to a loss of transcription factor binding site or a gain of transcription factor binding site. And TFs are involved in gene regulation, and if their binding behavior is changed, this might also have an effect on the target gene's regulation. So, how to detect regulatory SNPs and the affected TFs? To evaluate the effect of a SNP, one can compute a differential TF binding score. And given a wild type sequence and a mutated sequence, and a SNP, where here in this example a T is exchanged by an A, and we have a TF model, which can, for instance, be a PWM or a TFFM or also a SLIM model. And all these models describe the binding behavior of a TF. And the differential TF binding score is a log ratio which describes the difference in the binding affinity between the TF model and the wild type sequence and the TF model and the mutated sequence. And since we don't know the best position where the TF binds in the sequence, we basically slide the TF model along the sequence and consider all possible shifts where the TF model overlap the SNP. So we get several differential TF binding score shown here. And one possible strategy is, for instance, to keep the maximum one. Previous methods have been developed which all use different statistical approaches and to compute the differential TF binding score. And in this slide I shortly want to summarize them. And in general we noticed that not all of these methods provide a statistical significance of their results. So however, this is necessary to compare the results between different TFs. If they provide a p-value for their statistic, we notice that not all of them are independent from the TF model used. So, for instance, cubic PRET, which utilize in vitro universal PBM data, they derive their test statistic based on an ordinary least square estimation for the k-measures of their linear model. ECCB assumes that a score follows a multinomial distribution to model the PWMs they use. To the best of our knowledge, only the p-value computation in ECCB is independent from the TF model used. Unfortunately, they compute an exact p-value distribution to derive their p-value, which is extremely slow for large TF studies. Our aim is to derive a statistical approach that is not only fast, but also the p-value computation should also be independent of the TF model we use. So now you may wonder, okay, but how to compute the p-value then? And we derived a distribution which nicely approximates our absolute maximum differential TF binding score, which we denote as Dmax. Here I show you an example of observed Dmax values for a TF, which is actually zinc finger, and in comparison to our derived modified Laplace distribution. And as you can see here, the observed values really nicely can be approximated by our derived distribution. And since we now can know this distribution, we can compute a p-value for the Dmax score using the cumulative distribution function of the modified Laplace distribution. And this is extremely fast because it's just a lookup, we don't have to compute something, and the p-value computation is independent from the TF model we use. We benchmarked our statistical approach using SNP-Delix data, which is an in vitro measurement of the TF-DNA interaction for each allele of a SNP. From all the previously established methods, it was only possible to compare our approach to ECCB, since our approach also, like ECCB, only rely on the DNA sequence itself. And we run both approaches on the SNP data, and here in this precision recall plot you can see that both methods actually do perform really nicely, and where our method slightly improves on ECCB. And then we performed a runtime analysis where we used SNP sets of different sizes and also multiple threads, which are indicated here with this color coding. And you can see that ECCB, which is here denoted shown with these circles, is greater of a magnitude slower than actually our method. Based on the speed of our approach, we can now do analysis where one needs a proper random background statistic. And one example, for instance, is to determine cell type specific TFs, which are more often affected by the analyzed SNPs than one would expect on a random background set. And we did that for EQTLs for fibroblasts and lymphocytes downloaded from the GTEx portal. And in the scatter plot, you can actually see we mark those SNPs which are 2 times more often affected by the EQTLs than on a random background set. And for several of these TFs, we found evidence in the literature that they are associated to the analyzed cell types. In a second application, we worked together with the lab of Jaya Krishnan, and they are interested in identifying non-coding disease genes for cardiac vascular diseases. And to do so, we downloaded Jiva studies which are associated to cardiac vascular diseases, extracted their lead SNPs and the SNPs in LD. Then we used our statistical approach to predict the regulatory SNPs. And then we linked the regulatory SNPs to target genes using previously predicted enhancer gene interactions, and we downloaded them from the web server epigenomics. And in the lab, they are then decided for 40 promising non-coding genes, which they actually tested in vitro in a cardiac organoid model, which is a model that mimicked the human heart in disease and control conditions. And they identified that when they knocked out one of the non-coding genes, actually IgBP1P1, in disease conditions, that this affects the cell size and also the beating frequency in their heart organoid. And as a conclusion, they concluded that when one inhibited this non-coding gene, that this can improve cardiac function in cardiac vascular diseases. And this is actually my last slide. Here I want to shortly mention our software package, which is called SNEEP, where we combine our statistical approach, which is shown here, with epigenetic data and additional functionalities. And as I showed you, our statistical approach is really fast in comparison to other methods. And in the 2 applications, you already saw some functionalities of our package, but feel free to have a look by yourself, check it out. We have a GitHub repository for it and a Bioconner package. And if you're interested in more details of our statistical analysis, then you can have a look in our preprint, or you can step by to my poster. And then the only thing that's left to do is to thank our collaborators, Professor Jaya Krishnan at this lab, Professor Dr. Stefanie Dimmler for her support, and the whole Schulz lab. And with that, I want to thank you for your attention, and I'm happy to take questions.